We can make
So,
progress. Okay.
welcome to today's webinar with Cellusion, our innovation workshop. Just a couple of housekeeping for anyone who joins. Just make sure that your microphones are on mute and your cameras are off, and I will hand it over.
Thank you, Amanda. So, hello everyone. My name is Aurélien Petit. I'm the project manager for the Cellusion Clinical Development Department. Cellusion is a Japanese startup company that is currently developing regenerative medicine using iPS cells as the main source for manufacturing. And with this presentation, I wish to give to the audience an introduction about what our company is currently doing in terms of product development and explain, or at least try to explain how we aim to provide new treatments to patients suffering from corneal diseases where unmet needs remain despite all the technological progress observed in the past years. So before going forward, I just want to thank all the Genesis and the One Nuclear staff without who we will not be able to make this presentation today. So thank you guys to all the support provided. So first of all, as I mentioned just before, Cellusion is a Japanese startup company founded in 2015. We have around 30 people working hard within the wall, with almost 2/3 assigned to the research and development departments. So all of them keeping in mind this very important mission, which is regenerating the human potential. So Cellusion is committed to the development of regenerative medicine and improvement of the Japan presence in the global healthcare industry. We develop cell therapy using iPS cells, so induced pluripotent stem cells, as the main source for production. So pluripotent means that they can differentiate into any type of cells and proliferate indefinitely when we put them into culture. And so Cellusion has one vision: more freedom and more smiles to the world. You may wonder why we are talking about smile, whereas the presentation will focus on corneal diseases. So while it's true that our first product currently in development is aimed to treat patients suffering from corneal diseases, we expect in the near future to not only target ophthalmology, but also other therapeutics area that could benefit from our cell therapy technology. Here is our management team. So on the top left corner, the founder and actual CEO of Cellusion, the Dr. Shin Hato, so professor from the Japan Keio University with 20 years of experience as an ophthalmologist.
Sorry,
he conducted a
to just interrupt your slides. I don't think you shared your screen.
Oh, sorry.
That's okay.
It's strange. Because I was pretty sure that it was okay at the beginning.
The green, it was at the beginning, just the green button at the bottom.
Yeah.
Press share screen and then click on. There you go. Yep, that's working now.
That's working now? Perfect. Sorry for that. So yeah. First, so let me going back on that. First, the mission regenerating the human potential, and then a vision, more freedom and more smile to the world. Then here we have the management team. So top left corner, the founder and actual CEO of Cellusion, Dr. Shin Hato. Then I was mentioning on the right, Dr. Joji Hayashida is our head of business strategy. So he's an organic chemistry specialist, and he has also experience in investment evaluation in companies such as Astellas. On the bottom left, Takuya Kaneko is our head of the financial strategy. He has over 15 years of experience in investment banking activities, including IPO at Morgan Stanley. Then finally, on the bottom right, Nobuyuki Himai is our head of R&D. He's an experienced project leader for clinical development with several years of experience in Japanese pharma company. So here's the content we are going to explore together today. So first of all, without the past, it's difficult to have a present or even a future. So I wish to review quickly some historic fact regarding the development of treatment for corneal diseases. Then we will talk about the actual unmet needs the world is facing when we talk about corneal transplantation, keratopathy, which is the actual standard of care in the treatment of corneal diseases. Following that, we will be able to present the first regenerative cell therapy developed by Cellusion, codename CLS-001 or CECSi, and how we have characterized it in terms of potency purity, and safety. After that, we can have a look at some promising preliminary results observed in a Japan first-in-human study using CECSi. Then finally, we can talk about the current market, the competitor we have, and the future development perspective for Cellusion and regenerative medicine in general. But first thing first, during this presentation, I will say a lot about cornea, corneal endothelial cells, endothelium, etc., etc. So for some people, because some people may not be totally familiar with all of this, all those words, I just want to make a quick explanation regarding the eye's anatomy and how the mechanism of vision works. So at the beginning, there is light, and because without light, there is no vision. So then there is the cornea. The cornea is the transparent circular part of the front of the eyeball. Then when the light reaches the cornea, it is refracted into the lens of the eye, which then focuses it into the retina and the macula. Then the retina macula capture the information and it is sent to the brain thanks to the optic nerve. Then this information is translated into the actual vision. That's for the general process. But what is important for us today, the important part is the cornea. And what is the cornea? So the cornea is made of several layers. There is first the epithelium, which is laying on the Bowman membrane on the anterior surface of the cornea. There is the endothelium, which is laying on the basement membrane on the posterior surface of the cornea. Then between those two membranes, there is the stroma. And actually, the corneal stroma makes up for 90% of the corneal thickness. So it is a highly organized and dry environment. However, the stroma have the natural tendency to swell, which is not good for something that should remain transparent to ensure the correct vision. In terms of function, the endothelium is a monolayer of cells, so the corneal endothelial cells, organized in a characteristic honeycomb pattern, so with hexagonal shape. And it transports the water from the stroma into the anterior chamber of the eye, so the space between the cornea and the lens. This movement of water counters the natural tendency for the stroma to swell and is necessary to maintain the corneal transparency. I want to add that there is various diseases that affect the cornea and that may affect the functionality of the corneal endothelial cells and cause, indeed, transparency issues. But there is surgical procedure, the corneal transplantation, that can replace all or part of a sick cornea with the use of transparent healthy donor tissue. And lastly, because there is no blood vessel into the cornea, it is less prone to rejection compared to other organ transplants, allowing for the allogeneic corneal transplantation, so transplantation of another person's tissue, to be performed without taking care of any blood or HLA matching. So this is really important to understand this fact for what we will explain after during the presentation. A bit of history before going on further. So there is stories of eye or even heart transplantation in the old medieval text or old books. However, if we speak purely about the science, the original concept of corneal surgery can be dated back to the Greek physician Galen, so like 1800 years ago, something like this. So it is said that Galen have created what we call today the corneal tattoo. Today, it is sometimes done for cosmetic reasons, but back in the day, it was apparently to heal the scarring of the cornea that could lead to corneal opacity. But the real father of corneal transplants will be that guy here on the right. So the doctor Guillaume Peltier, a French ophthalmologist. So in 1789, he released a book that described the corneal disease classification and suggested a surgical procedure to remove an opacified cornea and replace it with an artificial device, looking like a lens. So it can be considered, in fact, as the first description of what is a full-thickness corneal replacement. However, there is no documentation available to suggest or prove that he ever performed such kind of operation. Then, on the 7th December 1905, the Dr. Edward Zirm of Czech Republic performed the first successful organ transplantation, and it was in fact an cornea transplantation. So the cornea of an 11-year-old boy who accidentally just died a few days ago was transplanted into the eye of a day laborer who was blinded by accident during his work. After the operation, the cornea did not get opaque even after the healing process, meaning no rejection was observed. After a few hours after the transplantation, the patient could see again, and he retained his eyesight for the rest of his life. So patient even returned to work. And the most important is that method developed by the Dr. Zirm is still the basis actually for the repair of corneal damages as of today. Then, few years later, there was the first successful transplantation, keratoplasty with a corneal tissue from a human corpse conducted by a Russian ophthalmologist, the Dr. Vladimir Filatov. In 1945, we have what we call the first World Eye Bank founded in New York in USA. So an eye bank is an institution responsible for collecting, harvesting, and processing donor corneas, and for distributing them to trained corneal graft surgeons. In 1949, the first keratoplasty performed in Japan by the professor Kitetsu Himaizumi. In 1956, the same professor established the first eye bank in Japan. In 1958, we have the first corneal transplant law announced in Japan. In 1963, I'm sorry for that, but I am a Beatles fan. I had to mention it. This is the first record of the Beatles. Please be released in UK. But what is interesting for us on this year is that this is the first licensed eye bank established in Japan. Licensing means that we can ensure standardization and consistent quality in the collection and preservation of a donated cornea. Then, in 2010, this is the last revision of the organ transplantation law in Japan, and we have actually 54 eye banks currently operating in Japan. So all of this, it brings us today, in 2023. The current treatment, the standard of care for corneal disease, is the corneal transplantation, as initiated by the Dr. Zirm. There is two types of corneal transplantation. First, on the left side here, the penetrating keratoplasty, or PKP, which involves replacing the anterior cornea, including epithelial, stromal, and endothelial layers. So the damaged cornea is removed, and then an healthy donated human cornea is transplanted. The advantage of this method is that because this is the oldest method for treatment, the procedure is well known, well established. So this is the advantage for this method. The inconvenience is that it's really invasive method with a high risk of immunological rejection and also other complications such as glaucoma or infection. The second type of corneal transplantation is called the endothelial keratoplasty. This type of surgery is performed to replace the endothelium when only the endothelial cells inside the cornea are affected. For example, there is a disease called the Bullous keratopathy. The patient's own endothelium is removed through a small peripheral incision, and then the endothelium from a donor cornea is transplanted. So the advantage of this method is that the corneal epithelium is not touched at all. There is no sutures and required, and there is less immunological rejection expected compared to the penetrative keratoplasty. But still, the intraocular pressure of the eye, intraocular pressure may increase due to a specific air pressure procedure required for the adhesion of the graft to the cornea, and there is still a risk of adhesion failure of this graft. So to sum up, the conventional corneal transplantation method, they are here, they are there, they are effective, but still there is still various complications with this method. In addition, so to perform corneal transplantation, as of today, donor corneas are still required. Secondly, corneal transplants are not easy for general practitioner, general ophthalmologist, and require the skill of an experienced doctor in order to be conducted. And third, banks are a necessary social infrastructure to ensure the safe and equitable delivery of donor cornea to the patients. So because there are few countries or regions in the world where all these conditions are met, there are a lot of patients on the waiting list for corneal transplant worldwide. So what I want to mention is that actually data from the GAINS report, so a global survey on corneal transplantation and banking, shows that there is approximately 13 million people worldwide waiting for a corneal transplant, while only 180,000 surgeries are performed each year due to a shortage in supply of donor cornea. There are various diseases that affect the cornea: keratoconus, bullous keratopathy, bacterial or viral infection, but more than half of the patients needing corneal transplantation suffer from what we call the bullous keratopathy, which is characterized by swelling and clouding of the cornea, resulting in the end in dysfunction, from the dysfunction and the decrease of corneal endothelial cell numbers. And this is ultimately leading to loss in visual acuity and or progressive blindness. So the bullous keratopathy can be caused by heredity, so due to a disease called the Fuchs corneal endothelial dystrophy that develop in patients at 50, 60 years old. Or bullous keratopathy can also be due to trauma caused by intraocular surgery, such as the cataract surgery, so surgery of the lens of the eye. The factor causing bullous keratopathy differ slightly by country. So for example, in Europe, UK, USA, the disease is predominantly hereditary, while in Japan and other Asian countries, it is often the result of complication from cataract surgery or other intraocular surgery. It should also be noted that in some country doesn't have this shortage issue, such as, for example, in USA, in United States, there is no shortage issue. And some other country try with new law regarding organ donation, such as the one in UK. They try to fight this. For example, in UK in 2020, a law was released, and now all the adults are considered donor when they die, unless they have a recorded decision not to donate. Then, in 2014, so Kyoto Prefectural University of Medicine began clinical research on cultured corneal endothelial cell transplantation, so in which corneal endothelial cells are cultured, enzyme treated to separate into single cells, and injected with ROCK inhibitor into the eye anterior chamber of patients suffering from corneal endothelial disease, including the bullous keratopathy we were mentioning just before. The ROCK inhibitor, I mentioned the ROCK inhibitor, this is a compound that can be involved in a lot of mechanisms and have many functions, but for the cellular level, it can help maintain the cell viability and the cell morphology. So thanks to this cell injection during this study, patients recovered transparency and normal thickness of their cornea. So this specific trial, demonstrated the usefulness of the cell transplantation for corneal disease, as a treatment for corneal disease, such as the bullous keratopathy. So the major advantage of cell transplantation therapy is that it is minimally invasive compared to conventional corneal transplantation. And another advantage will be that conventional corneal transplantation can be performed as a backup treatment for non-responsive case or recurrent cases. So it is expected that in the future, cell therapy may become a first-in-line treatment. But the problem here with this particular study and this particular technology is that they are still dependent from donor cornea and from a bank in order to manufacture this cell suspension. So what if a regenerative cell therapy for corneal disease, which is not dependent on A-banks, could be developed. So one solution will be to use the iPS cells. Several methods to make corneal endothelial cells from induced pluripotent stem cells, so iPS cells, have been recently reported in various papers. It has been reported that the corneal endothelial cells originate from what we call here the neural crest cells, or NCC. The NCC are precursor to a wide variety of tissue, such as the bones, brain, eyes, and therefore one method to obtain the corneal endothelial cells by iPS cell culture is called the indirect differentiation induction, using this NCC intermediate stage. However, during their research, so the Dr. Shin Hato and his team tried to determine the optimal timing for corneal endothelial derivation, and they discovered that corneal endothelial-like cells were most efficiently produced by direct induction from iPS cells and omitting this NCC differentiation. This is what we call the direct differentiation induction process. So the corneal endothelial-like cells derived are considered, so we can derive directly from iPS cells corneal endothelial-like cells, but those corneal endothelial-like cells derived are considered a little bit different from the human corneal endothelial cells because we don't have this NCC intermediate stage. So in addition, there is no established marker specific to corneal endothelial cells, making their characterization really difficult. Therefore, it was proposed to call those cells, these cells, obtained by direct differentiation induction, corneal endothelial cells substitute derived from iPS cells, or CECSi cells, and codename CLS001. Then, in January 2015, the Professor Tsubota, Shinmura, and Hato from the Keio University founded Cellusion in Tokyo, and Shin Hato was appointed as the CEO. So we have CLS001, the corneal endothelial cell substitute derived from iPS cells, or CECSi. So using iPS cells generated from skin, blood, other sources from healthy individuals, Cellusion succeeded in producing CECSi cells, which function in a similar way as human corneal endothelial cells. So CECSi cells are believed for having considerable potential as an effective treatment for bullous keratopathy by restoring the normal thickness and transparency of the cornea with a single injection into the anterior chamber of the eye, leading to cell adhesion to the posterior surface of the cornea. So we believe the technology and product that Cellusion is developing can solve the bottleneck in cornea transplant, so the shortage of donor cornea. First, it is possible to mass produce CECSi cells with a standardized quality because we use iPS cells for production. Secondly, CECSi cells is a cell therapy provided to the patient with a single injection into the eye, meaning the procedure is less invasive and less stressful for the patient, and that ophthalmologists can learn the surgical techniques in a short amount of time, leading to a procedure that could be widely accepted quickly. Then, the last point, unlike other donor-dependent therapies, the product we are developing always use the same allogeneic iPS cell stock, meaning that they can always be delivered to patients in the same way as any ordinary pharmaceutical product, even without an A-bank. Lastly, in addition, it is possible to cryopreserve those cells and at the final stage of production, which means that we can produce and stock them in large quantities in advance, and we can deliver them as necessary. So the conventional corneal transplantation method still presents a lot of problems, various complications, but with CECSi cells, we have a cell therapy that is potentially effective treatments for bullous keratopathy, with reduced burden for the physician, for the general ophthalmologist, because this is an easy-to-use treatment, And we have reduced burden for the patient because the cell injection procedure is less invasive compared to regular transplantation. The wound is so much smaller, and risk of complication decrease with the lower invasiveness of a procedure. The only downside for the patient would be that you have to stay 3 hours in a prone position, so face down position, in order to allow the cell to settle in the posterior surface of the cornea. That's the only downside. So by now, you normally have a good understanding of what is the CECSi cells, but you also heard that I mentioned we don't consider them as regular human corneal endothelial cells because we use a new differentiation process omitting the neural crest cell stage. That's why we said corneal endothelial cell substitute derived from iPS cells. So first, regarding the production process, one batch of production will take about 1 month, with the expansion procedure for the iPS cell, and then the induction differentiation procedure to obtain the CECSi cells. The culture medium used during the whole production process is chemically defined animal human origin free. And then, from there, how do we know how to ensure that our cells works in the same way as human corneal endothelial cells? So simply said, how do we confirm the quality of our product? So we do it with the observation of functional corneal cells marker. And we compare the level of expression of those markers in the CECSi cells with what can be observed in a reference cultured human corneal endothelial cell line. So it's a line well used in the research, which is called the B4G12 cell line. We selected 3 functional markers as reference tests to ensure the quality of our cells. First, the encadherin. So encadherin to confirm the barrier function of the monolayer of endothelial cells, which will selectively restrict the flow of liquid from the anterior chamber to the corneal stroma, Then we have the sodium-potassium ATPase and the mineralocorticoid receptor to confirm the pump function of the cells, and therefore the exchange of fluids from the stroma to the anterior chamber. By conducting flow cytometry analysis for those 3 markers, we observed similar high level of expression between CECSi cells and the reference cell line, meaning that we can expect CECSi to work in a similar way and with a same active function that will help maintain the corneal transparency compared to human corneal endothelial cells. In addition, we also perform immunostaining of corneal endothelial markers, such as the ATPase mentioned previously, and also encadherin, and we confirm their presence into our cells, into our CECSi cells, and therefore the possibility to use CECSi as a treatment, as a replacement for human corneal endothelial cells. Some tests were also conducted to ensure the purity and the safety of CECSi cells. So firstly, the research of the undifferentiated cell marker OCT4. So OCT4 is specifically expressed in all kinds of pluripotent cells, like the iPS cells. It is a required marker to establish and maintain the cellular pluripotency. When the cells reach their final stage of development, they lose their pluripotency, so OCT4 is not expressed anymore. So to confirm our CECSi cells differentiation process is completed, we observe OCT4 marker by immunochemistry. So iPS cells express OCT4, CLS001 doesn't, as we can see here. We then perform a more sensitive test, qualitative PCR analysis of OCT4 expression in CECSi cells compared to reference cell line spiked with different percentage of iPS cells. And as you can see on the graph here, for CLS001, so the CECSi cells, they were way below the limit of detection for this method, which is considered to be 0.01%. Lastly, we wanted to confirm the absence of abnormal proliferation of the CECSi cells, as for regular corneal endothelial cells, because they don't proliferate naturally. So for this, we use the spiking by HeLa. So HeLa is a well-known cell line used in scientific research, perfect to evaluate the abnormal proliferation of cells. So during the experiment, CECSi cells were spiked with the indicated number here of HeLa cells, 3 flasks by group. And what we have seen is that CECSi cells growth was low during all the study, during all the analysis, while the HeLa spike groups showed abnormal increase in cell proliferation. So with this experience, we confirm that CECSi cells don't proliferate and don't proliferate abnormally. Then we were able to conduct our first animal preclinical study. So the purpose of the study was to confirm the efficacy and the toxicity of CECSi cells as a proof of concept study in a Bullous keratopathy cynomolgus monkey model. So meaning that we induced artificially a detachment of corneal endothelial cells by abrasion using a needle into the eye of monkey. So CECSi was injected into the anterior chamber of 6 female monkey, and we had also a control group where only a culture medium used as a substrate for transplantation was injected into 6 female monkey. And we observed the results up until 4 weeks after transplantation. So, as you can see, improvement of corneal edema was confirmed up until 28 days following the cell injection into the CECSi group. On the 6th monkey with CECSi transplanted, we observed a successful transplantation of the CECSi cells that grew on the posterior surface of the cornea, exhibiting the corneal cells morphology with their tight junction. Immunostaining of the USP9 marker was used to distinguish the transplanted human male CECSi cells from female monkey study. Why? Because in fact, to manufacture CECSi cells, those cells were prepared using iPS cells originated from a male donor and therefore express specifically this USP9Y marker. So the transplanted group was positive for USP9Y, whereas the control group was negative. Result shows that we have reduced corneal edema, recovered corneal thickness, therefore recovered corneal transparency. That also could mean help in recovering the visual acuity. So everything was not perfect on this study. There is limitation to this study. It is true that we observed a case of immunological rejection on 3 monkeys, but we have to consider that the monkey Bullous keratopathy model is a more severe model compared to the procedure to be used in the human clinical setting. In the monkey Bullous keratopathy model, all the cells are detached, whereas it is limited to a small area of less than 8 mm in diameter when doing the procedure in the human clinical setting. Nevertheless, the fact that the CECSi transplant group showed a significant therapeutic effect within 1 month observation period compared to the control group suggests the efficacy of the cell therapy, of the CECSi cell therapy. The next step then was to launch a first-in-human study. So an investigator-initiated trial supported by Cellusion was launched in the Keio University in Japan in July 2022. So the objective of this study was to evaluate the safety and efficacy of CECSi in the severest case of Bullous keratopathy in human. Severest case or severe case means relapsing case of Bullous keratopathy after receiving a previous penetrative keratoplasty. It was a single cohort, single dose, open label, and 52 weeks follow-up study on 1 patient. And a dose of 8 cross 10 to the 5 cells for 160 microliters of cell suspension with ROCK inhibitor was injected into the anterior chamber of the eye of the patient. So today, I can disclose the 3-month safety and efficacy data for our first treated patients. A paper will be published later next year with the 1-year study results, but at this point, this is still confidential material. I can just say that the results were good. But to come back on the case, so the patient was a 73 years old male. Primary disease was keratoconus. He received the first penetrative keratoplasty 48 years ago. But after that, the corneal endothelial cells gradually decreased and bullous keratopathy was observed. And therefore, the patient received another penetrating keratoplasty at the Keio University Hospital 6 years ago. He also had cataract surgery after that with good result, but unfortunately, in 2019, he developed bullous keratopathy again and then requested to participate in this clinical study. In this clinical research, baseline examination showed very poor visual acuity, intraocular pressure within normal ranges, bullous keratopathy, of course. You can see here opacity of the cornea just below the center, probably due to an infection of the scar of the cornea, but there were no other abnormality observed at this time. Then the central corneal thickness was measured as 863 micrometers by OCT, which is way higher than the normal ranges for the corneal thickness. So after 3 months, in terms of safety, no abnormalities, worsening detected, and no abnormal cell cluster detected into the anterior chamber of the eyes. No adverse event reported during the surgery, and no adverse event during the follow-up period, up to 3 months. And the intraocular pressure at 1, 2, and 3 months after the cell transplantation was measured as within the normal ranges. As for the efficacy, as you can see here on the slit lamp microscopy photography, after 3 months, 3 months after transplantation, we can observe a good improvement in corneal transparency. Just on the lower part of the cornea, so as mentioned previously, there is still this moderate opacity that remains that can probably be explained by the pre-existing corneal scar, as I mentioned previously. Then, in terms of corneal thickness, so the OCT evaluation shows gradual decrease every month and reaching value of 611 micrometer at 3 months after cell transplantation, which can be considered within the normal ranges for corneal thickness, while being on the upper range. Finally, for the best corrected visual acuity, the improvement remained limited. Again, probably due to this infection of the scar over there, but the patient noted himself an improvement in brightness. So this is a subjective symptom. So to sum up, a first-in-human study for CECSi cell suspension, CLS-001, transplantation was conducted at the Keio University in Japan. No intraoperative or post-operative adverse events were observed, and therefore, the IDMC, so the Independent Data Monitoring Committee, confirmed the safety of the product at 3 months data review point and approved the study to be continued. Improvement of the best corrected visual acuity, central corneal thickness, minimal corneal thickness have been observed up to 3 months. And now the follow-up until 52 weeks after transplantation results are expected to be published during the first half of 2024, so next year. So now, knowing all those previous results, we can move to the presentation of the global development plan for CLS-001. So at the top, you can see the Keio investigator-initiated trial, which was completed a few months ago and for which we will have data next year. Then we have the launch during the second half of 2024 of the sponsor clinical trial in Japan. So it will be a combined phase 1-2 study to evaluate the safety and efficacy of CLS-001 in the Japanese population. In parallel, we want to launch a US phase 1 in 2025 to cover the safety and preliminary efficacy in the US population. Then, using data of those 2 studies, we wish to continue our development with a global phase 3 study extended to Japan, US, UK, Europe, why not China also. And in Japan, PMDA, so the regulatory authority, the PMDA allows a conditional approval after the end of the phase 2 clinical trial for regenerative medicine. That's why you can see here conditional marketing authorization in 2026 in Japan. Let me come back to this point a bit later during the presentation. So in terms of positioning, Cellusion aims to treat the disease called the Bullous keratopathy, a disease that represents the grade 4 of the corneal endothelial dysfunction, according to the severity classification made by the Japanese Ophthalmological Society. Expected target population will be the mild and moderate patients, meaning in fact patients with a primary disease as Bullous keratopathy, and not a relapsing case of Bullous keratopathy following a previous penetrative keratoplasty. It doesn't mean that we cannot extend to this severe population later, or even to grade 1 to 3 of the corneal endothelial dysfunction in the near future. But for the moment, we just focus on the mild and moderate patients. We want CLS-001 to be positioned as a first-line treatment, so even before any kind of standard corneal transplantation, that could therefore be used after at the second-line and third-line therapeutic option. In terms of production for CLS-001, we plan to manufacture and be able to treat 1,000 patients per batch. The production of CLS-001 will take 1 month, including culture and expansion of the iPS cells and induction of the direct differentiation into CECSi cells. Then the cells can be cryopreserved and delivered to medical institutions all over the world. We have 6 patents at the moment that cover our production process, including the specific culture medium necessary to induce the direct differentiation of iPS cells into CECSi cells. We estimate that Cellusion is today the most advanced company for the development of cell therapy using iPS cells as source material in the treatment of corneal diseases, because we will soon enter in our sponsored phase 1 stage of development. If we have to name one major competitor, it will be, I think, Orion, with their new product Visnova, that recently got the PMDA conditional approval in Japan, I think in March of this year, if I'm not mistaken. They indeed have a similar product. It's cultured corneal endothelial cells, but they still rely on the donor cornea in order to be able to manufacture it. So for this particular reason, Cellusion CLS-001 still keep an interesting competitive advantage, as we can solve the corneal donor shortage issue by using the iPS cells as a source for the manufacturing. The global market for corneal transplantation is actually driven by 3 major factors. So the aging population, older people are subject to higher incidence for the corneal disease, apparition recently of the intraocular surgery, for example, the cataract surgery, and one of the complications is the apparition of disease like the Bullous keratopathy, and in general, the growing population in the world. So these 3 factors can explain the potential global growing market for the corneal transplantation. This global market is projected to grow from $421 million in 2021 to $641 million by 2028. So although the increasing incidence of eye diseases is driving the market, the risk associated with the regular corneal transplantation and the shortage of donor cornea are expected to limit a bit this market growth. This is where our solution become interesting, because we want to solve the shortage of donor corneas, and for product, we expect to reduce the complication normally associated with corneal transplantation. Then one last thing is that of course, the 3 bigger markets at this moment are the US, UK and Europe, and the Asian country, including Japan. So our plan actually is to accelerate the development in Japan using at our advantage the recent Pharmaceutical and Medical Device Act on regenerative medicine, and target, as mentioned previously, conditional marketing authorization approval in Japan in 2026. That will be followed by other areas such as US, UK, Europe in 2029 and 2030. So to come back on this, because this is one of the key factors for the Japan development, In Japan, since November 2014, due to a huge demand for new regulation towards the regenerative medicine, products distributed by companies and clinical trials are regulated under the Pharmaceutical and Medical Devices Act, so the PMD Act, so on the right side of the screen over there. In addition to this one, there is the clinical research. So for clinical research, other than clinical trials intended for submission to get approval, this kind of clinical research, they are covered by what we call the Act on the Safety of Regenerative Medicine, so the RM Act. So clinical research under the RM Act and clinical trials under the PMD Act have different data integrity standards. Clinical research are approved by the Ministry of Health, Labor and Welfare in Japan, while the clinical trials, they must be approved by the PMDA, the Drugs Regulatory Authority, with a specific clinical trial notification. Good clinical practice only applies to the clinical trials. But for clinical research, data may not be considered as reliable data for approval review under the PMD Act, unless they fulfill this GSP standard, even if it's not required. So, however, exploratory clinical research under the RM Act can be used to design confirmative clinical trials. And in the early clinical development phase of regenerative medicine, products in Japan are often first evaluated with this clinical research pattern, which is in fact considerably cheaper than clinical trials. And if the prediction of success is high, then a company-sponsored clinical trial may be initiated. This is in fact the pattern that has been chosen by Cellusion, with conducting first a first-in-human study, a clinical research in the Keio University, and then moving to the phase 1-2 sponsored clinical trial. What is important is that thanks to the PMD Act, there is the possibility in Japan to get the conditional and time-limited approval for regenerative medicine product based on limited efficacy data such as surrogate endpoint data collected during a phase 2 clinical trial. So it's in fact similar to what we have in US with the FDA accelerated approval. For this reason, Japan is one key market when considering developing a new regenerative therapy, regenerative medicine. This is also why Cellusion, being a Japanese company, may not be only interested in licensing out our product, but also licensing in for potential Japanese developments. After a conditional and time-limited approval, of course, the phase 3 conduct remains mandatory in order to confirm the safety and efficacy further for the patients, and it is required to apply for regular approval within maximum 7 years after the conditional approval. That will be my last slide, I think, and with some take-home message. So, Cellusion, we want to be pioneer of iPS cell-based regenerative medicine for treatment of corneal diseases. We can mass produce the CECSi cells, the CLS001, from iPS cells while ensuring stable quality and cryopreservation of the cells. We aim to treat Bullous keratopathy patients from all around the world with our cells. Thank you for your attention today. We have a stand in the atrium, so this is time number 9, so please do not hesitate to come to discuss about our company, about Cellusion. We may also discuss maybe about the Beatles. I mean, whatever is fine, so please do not hesitate to come. Thank you.
So we actually came up with a couple questions for you. Can you see the chat box at the bottom?
Let me check. I see. Okay. Hey man, thank you for joining. Please pop in question. Okay. Is there any particular advancement or improvement expected in CECSi treatment for Bullous keratopathy in the near future? What makes someone a good candidate for CECSi instead of a corneal transplant, and what are the differences between the recovery time of the two? Let me think. So the first question, are there any particular advancement or improvement expected in CECSi treatment in the near future? Advancement or improvement. I'm not sure to exactly get the question. Does it mean that we're getting all clinical development plan or new formulation?
Yeah, I guess
or...
however you take the
Um,
question.
Yeah. So as I mentioned, we already conducted the first in-human study, and now the next movement, the next development will be to launch our first in-human phase 1/2 clinical trial in Japan. Having said that, I mean, because we are still at the beginning of the process, still at the beginning of the development, it is known that during the course of the development, there is still improvement that can be made during the manufacturing process. So at this moment, I cannot answer exactly for reading what kind of improvement can be done because I'm not a CMC expert.
Right. Okay.
But it is sure that before launching the product, between the time we launch the product in Japan and after when we finish to conduct a phase 3 for a global launch, there is definitely improvement that can be made maybe to the formulation in order to make it even more efficient. That's for sure.
Okay.
And the second question would be, what makes someone a good candidate for CECSi instead of corneal transplant, and what are the difference between the recovery time of the two? I don't have specific data for the recovery time, but as I mentioned, what will make someone a good candidate for CECSi instead of corneal transplant? Before going to any kind of corneal transplant, the cell transplantation will be better because it's less invasive. So any candidate will be a good candidate, in fact, as long as we observe a corneal disease. So it can be, as I mentioned, the Bullous keratopathy, but also when I mentioned, for example, the target population, we know that even before reaching the Bullous keratopathy stage, there is also the what we call the grade 1 to 3, so which is in fact the beginning of the first corneal endothelial dystrophy. So maybe even such kind of patient with a small corneal dystrophy could benefit for CECSi, but in order to confirm that, we need also to make clinical trials.
Right. Okay. That's cool. Okay. Well, if you don't have any more points, we will see you tomorrow in person.
Yes.
Thank you very much for
that.
today. Catch up on your sleep.
Yeah, I will try.
Okay.
Thank you. Thank you for the
Thank you very
introduction.
much. Bye.
Bye. See you next time.